Welcome back, everybody, to another deep dive.
Yeah.
Today, we are going to be tackling a really fascinating case
Oh, yeah.
of neuronal ceroid lipofuscinosis (NCL) in a Schapendoes dog.
Yeah.
This is a paper by Bellamy and colleagues, just published in Animal Genetics in 2024.
Very recently.
Just out, and we're going to be diving into this case,
Yeah.
looking at the clinical presentation, the diagnosis, the genetic basis, and potential implications of this newly discovered NCL variant in Schapendoes.
Right. And I think this is really interesting because NCL is also known as Batten disease in humans.
Oh, right.
And so we see it in people and we see it in animals, and we're always trying to figure out what is the cause.
The cause, and how can this help us treat either dogs or humans?
Yeah, and this particular paper I think is fascinating because they really dove deep into this one dog's case, and they were able to find the genetic cause.
Absolutely.
Which is really cool.
Yeah. So it's not just about one dog
Right.
that has implications for
Exactly.
Schapendoes breeders, and maybe even for NCL in humans.
For sure.
So let's
Okay.
I'm ready to dive in. What about you?
Let's dive in. I am ready.
Okay.
So NCL, just to kind of step back a little bit, is a group of inherited neurodegenerative disorders.
So
It affects the nervous system,
Yeah.
and it gets progressively worse over time.
Right.
Sad.
Sad, and known for its early onset as well.
Yeah.
And oftentimes a shortened lifespan,
Yeah, for sure.
both in humans and animals.
Both in humans and animals.
Yeah.
And so there are currently 13 different NCL genes identified,
Okay.
and each one causes a slightly different form of the disease.
Okay. So there's like a spectrum.
There's a spectrum,
in clinical science.
Yeah. So what makes this particular case so interesting in this Schapendoes is that they found a novel variant that's not been described before. So it's potentially adding another piece to the NCL puzzle.
Wow.
Yeah.
Okay. Well, let's talk about this dog.
Okay.
So it was a 19-month-old male Schapendoes brought to the Norwegian University of Life Sciences.
Yep.
And he was already showing a lot of concerning symptoms by this point.
Absolutely.
Yeah.
And actually his decline seems to have started when he was about 12 months old.
Wow.
So just think about that. He's barely a year old and already starting to face some serious neurological challenges.
Yeah, that's really young.
Yeah.
For sure.
It's heartbreaking.
Yeah, it's heartbreaking. The article lists a whole cascade of symptoms.
It does.
Difficulty climbing stairs, progressive vision loss, leading to blindness, problems with balance and coordination.
Right. Ataxia.
Ataxia, they call it in the paper.
Yeah.
And even tremors.
Yeah, and it doesn't stop there.
Wow.
He also lost some learned behaviors, started having episodes of myoclonus.
So those are...
which are these involuntary muscle jerks.
So, gosh.
And he even experienced periods of unresponsiveness, especially at night.
Wow. So this is...
because it's a lot.
Yeah, a lot for a young dog to deal with.
Yeah, absolutely.
So how did they
diagnose this dog with NCL?
Yeah, so they started with a thorough neurological exam.
Okay.
And that probably involved testing his reflexes, his gait, his mental state.
Okay.
And then they ran a battery of tests.
Okay.
Including blood work, urinalysis, and even a metabolic screen.
Okay, so they were trying to figure out what was going on.
Yeah. So what did they find?
So they did find some abnormalities in the blood and urine tests.
Okay.
Things like neutropenia, which is a low white blood cell count, and aminoaciduria, which means that there were amino acids in his urine.
Okay.
But these findings alone couldn't pinpoint the cause of his neurological decline.
Yeah. So how did they finally confirm the diagnosis?
So sadly, because of the severity of his condition, he was euthanized.
Okay.
And this allowed them to perform a full necropsy.
So necropsy is like
basically an autopsy for animals.
Autopsy for animals.
Yeah.
Okay. And so what did they find in the necropsy that pointed to NCL?
Okay. So in the necropsy, they found evidence of cerebellar atrophy.
Okay.
And that means shrinkage of the cerebellum.
Yeah. The cerebellum is the part of the brain that controls coordination and movement.
Right. So that makes sense.
Right.
Considering all the problems that this dog had with balance and coordination.
Right. And those episodes of myoclonus.
Exactly.
Yeah.
But even more telling, they found these characteristic cytoplasmic inclusions.
Okay, hold on. What are those?
Within the neurons of his brain.
Yeah. What are those?
Okay. So think about neurons as like the communication highways of the brain.
Okay.
They're sending signals back and forth to control everything we do.
Right.
These inclusions are basically abnormal clumps of stuff that build up inside the neurons, disrupting their normal function.
And why are these inclusions specifically linked to NCL?
So these inclusions have very specific characteristics.
Right.
They were PAS positive, Sudan black positive, and they showed autofluorescence under the microscope.
Okay.
And those are all classic hallmarks of NCL.
I see.
Yeah.
So finding these inclusions in his brain tissue was a pretty strong indicator that he had NCL.
Absolutely. And they went a step further and used electron microscopy.
Wow.
And this allowed them to look at the ultrastructure of those inclusions in even greater detail.
Wow.
So providing even more confirmation of that diagnosis.
So it's like looking under a really powerful magnifying glass.
Exactly.
To be able to see those.
To see those tiny little details.
Yeah.
But the big question remained, what was the genetic cause behind all of this?
Yeah, that's what I was wondering. How did they go about finding the genetic culprit?
Okay, so they started by analyzing the dog's pedigree.
Pedigree, that's like a family tree?
Yeah, like a family tree for dogs.
Yeah.
And this suggested that the mode of inheritance was recessive.
Okay, can you explain what that means?
Yeah, so recessive inheritance means that both parents need to carry the faulty gene for the disease to manifest in their offspring.
Okay.
So if only one parent carries the gene, the puppies won't develop the disease, but they could still be carriers themselves.
Okay, that makes sense.
Yeah.
So how did they actually pinpoint the specific gene responsible?
Oh, okay. So they use a really powerful technique called whole genome sequencing.
Wow.
And this basically allows them to read the entire genetic blueprint of the dog looking for any abnormalities.
Wow. So what did they find?
And they hit the jackpot.
Okay.
They discovered a novel missense variant in the CLN6 gene, and specifically a T to C change at a very precise location on chromosome 30. And this change leads to a substitution of one amino acid for another in the CLN6 protein.
Hold on. Can you back up a second?
Yeah.
What exactly does a missense variant mean?
Okay, so think of it like this. Genes are like recipes for proteins, right? Yeah. And proteins are the workhorses of our cells. They do all sorts of important jobs.
Right.
A missense variant means that there's a typo in the recipe.
Okay.
One letter in the DNA code is changed, which can result in a different ingredient being added to the protein. In this case, a different amino acid.
Okay, so that throws off the whole recipe.
Exactly.
Yeah.
It can mess up the protein's function.
Okay.
And what's really interesting is that they didn't find the specific CLN6 variant in any of the dog's healthy siblings, nor in a bunch of other healthy Schapendoes, or even in dogs from 35 other breeds.
Oh, wow.
So it's looking pretty likely that this CLN6 variant is the root cause of NCL in the Schapendoes.
Yeah, so this is a pretty big discovery.
It is.
Right.
It is. It gives us a better understanding of NCL in dogs, but it also has potential implications for human NCL research. But we'll delve deeper into those aspects a little later.
I'm definitely intrigued. We've uncovered the genetic culprit.
Yeah.
Established a strong link to NCL.
Yes.
But what does this mean for Schapendoes breeders and for potential treatments for NCL?
That is a great question.
Yeah.
So now that we know the specific genetic variant, breeders can start using DNA tests to identify carriers and prevent them from being bred together.
So that's huge.
Yeah. So this can help to reduce the incidence of NCL in future generations of Schapendoes.
Right. That's a huge win for the breed, for sure.
Yeah, absolutely.
But what about treatment? Is there any hope?
That's the burning question, isn't it?
Yeah.
Unfortunately, there's no cure for NCL yet. But this discovery could pave the way for new therapies.
Okay.
So by understanding the precise genetic defect and how it disrupts cellular processes, researchers can start to develop targeted treatments that address the root cause of the disease.
Yeah.
Yeah.
So this Schapendoes dog could be a key player in advancing NCL research for both animals and humans.
And it highlights the interconnectedness of veterinary and human medicine.
Absolutely.
Yeah.
Like what we learned from our animal companions can often shed light on human health.
For sure.
And vice versa.
Exactly.
Yeah, that's powerful.
It is.
But we're just scratching the surface here. There's a lot more to explore about the CLN6 gene and how this specific variant disrupts cellular function.
Exactly. So in the next part of our deep dive, we're going to delve into the intricate workings of CLN6.
Okay.
And the cellular chaos caused by this missense variant.
All right. I'm ready for it.
Stay tuned.
Right.
Welcome back.
Okay, so we know that this CLN6 gene is messed up.
Right.
But what exactly does CLN6 do in a healthy dog or even a healthy person?
So in a healthy dog or healthy person, CLN6 provides the instructions to make a protein called ceroid lipofuscinosis, neuronal protein 6.
Catchy.
Yeah, but we're just going to call it CLN6.
CLN6, got it.
Yeah, and this protein lives within the endoplasmic reticulum, or ER.
Okay, the endoplasmic reticulum?
The ER.
Is that like some kind of brain structure?
Not quite.
Okay.
It's actually a network of membranes found inside almost every cell in your body, and the dog's body, too.
Okay, so it's busy.
It's like a manufacturing and transportation hub.
But what does it manufacture and transport?
Proteins. Yeah, so the ER is crucial for protein synthesis, folding, and transport. Okay. So to basically make sure that the proteins are made correctly and delivered to where they need to go within the cell.
Wow.
Yeah.
Okay, so where does CLN6 fit into all of this?
So CLN6 is like a chaperone.
Okay.
It ensures that specific enzymes that are destined for the lysosomes are properly packaged and shipped from the ER.
Okay, so lysosomes, those are the cell's recycling centers, right?
Exactly.
Okay.
They break down.
Yeah, they break down cellular waste and other unwanted materials.
Right.
Yeah.
Okay.
So now, if CLN6 is mutated like it is in this Schapendoes, this transport system kind of breaks down.
So then those necessary enzymes can't get to the lysosome.
Right. They can't reach the lysosome.
Where they're needed.
Where they're needed.
Okay.
Yeah. So then what happens?
Yeah, what happens then?
Well, the waste materials and cellular debris start to accumulate, causing damage and dysfunction.
So it's like a traffic jam.
It's a traffic jam. Yeah.
Yeah.
And in NCL, this is happening in neurons.
Right. Those really important cells.
Right.
The all-important cells responsible for transmitting information throughout the nervous system.
So it's like a domino effect.
It is like a domino effect.
The mutated CLN6 gene disrupts the protein transport, leads to lysosome dysfunction, and then ultimately neuron damage.
Exactly. And remember, it's not just any mutation in CLN6. We're talking about a specific missense variant.
Right, the T to C change.
Right, the T to C change.
That swaps a leucine for a proline.
Exactly.
In the CLN6 protein.
Yeah, and you mentioned earlier that proline is known to cause kinks in a protein structure. So how does that play into this? Well, amino acids are the building blocks of proteins. Right? And their specific sequence determines the protein's 3D shape and ultimately its function. So introducing a proline can significantly alter that shape.
So it's like trying to fit a square peg in a round hole.
Yeah, it's a good way to put it. Yeah, so basically this missense variant is disrupting the delicate balance within the cell, hindering lysosomal function, and contributing to those devastating neurological symptoms.
So a single letter change can have huge
consequences.
Yeah, it highlights the importance of understanding the genetics behind these diseases.
Absolutely.
But remember, it's not just about this one Schapendoes. This finding has potential implications.
For human.
For human NCL6 research as well.
So how does this dog's case help us understand human NCL?
Well, as we've discussed, NCL affects both humans and animals.
Right.
So by studying this naturally occurring canine model, we can gain valuable insights into the disease mechanisms that might be applicable to humans as well.
So this Schapendoes could hold the key to unlocking new therapies.
Potentially, yeah.
Or even a cure for this devastating disease.
Right. The humans, while this research provides crucial information about NCL6, it also raises some important questions.
Like how prevalent this specific CLN6 variant is in the broader Schapendoes population.
That's one. We need more research to determine the carrier frequency and understand the true extent of this mutation within the breed. It's also critical to investigate how this form of NCL manifests in dogs compared to humans.
Right, because while there might be these parallels in terms of the underlying genetic defect. There might be nuances.
Exactly, in the clinical presentation or disease progression that are specific to each species.
Right.
And that's where further research using this Schapendoes model comes in.
Absolutely.
It could provide valuable insights into the complexities of NCL6 and potentially lead to more effective treatment strategies for both dogs and humans.
It seems like every answer leads to more questions.
It does.
But those are good questions to have.
They are good questions to ask.
They point us towards the next steps.
They do. And those next steps are what we'll explore in the final part of our deep dive.
Okay.
We'll look at the broader implications of this research, from veterinary diagnostics and breeding practices to advancements in human NCL6 research.
All right, I'm ready.
Stay with us.
Welcome back to the final part of our deep dive into NCL.
Yeah.
We've covered a lot of ground.
We have.
You know, from the heartbreaking symptoms of this Schapendoes dog to the intricate workings of the CLN6 gene and just the havoc a tiny genetic variant can wreak on a cellular level.
It's incredible.
Yeah, it's incredible.
Yeah.
But before we wrap up, I think it's important to kind of zoom back out and look at, you know, the wider impact of this research.
Absolutely. I think this case is a prime example of the interconnectedness between veterinary and human medicine.
Right. Like what we learn from animals, it can often shed light on human diseases.
For sure.
And vice versa.
And vice versa.
Yeah, like a two-way street.
Yeah, two-way street of knowledge sharing. So this discovery has like immediate and practical implications for Schapendoes breeders.
Yeah, so now that the genetic variant has been identified, a DNA test can be developed to screen breeding dogs.
Yeah, so breeders can make informed decisions about which dogs to pair.
Exactly.
And potentially prevent future puppies from being born with this form of NCL.
It's huge.
Yeah, that's huge. Yeah. And then the benefits go beyond the Schapendoes breed, right?
Basically, by studying this naturally occurring canine model, we can gain valuable insights into human NCL6 as well.
So it's amazing to think that this one dog could contribute to developing new therapies or even a cure for NCL, not only in dogs, but potentially in humans too.
It's certainly a beacon of hope.
Yeah.
Researchers can use this model to study disease progression, test potential therapies, and really dig into the underlying mechanisms of the disease.
That's the crux of the matter, isn't it?
It is.
How can we translate these findings into tangible treatments?
Exactly.
And so, you know, this Schapendoes case opens up so many exciting avenues for future research.
It underscores the importance of collaboration between veterinary specialists, geneticists, and researchers from diverse fields.
Right. It's a true team effort.
It is a true team effort.
To tackle complex diseases like this.
For sure.
And the more we understand about, you know, the genetics and the cellular mechanisms involved, the closer we get to finding effective solutions.
Right.
Well, I think we've successfully navigated the complexities of this case.
I think so.
And gained some truly valuable insights.
Oh, I have.
Along the way.
Yeah. We've seen how a single letter change in the DNA
can have a ripple effect.
Yeah.
Disrupting essential cellular processes.
Yeah.
And leading to devastating neurological consequences.
We've learned about the intricate workings of the CLN6 gene.
Yes.
And its critical role in maintaining that delicate balance within our cells.
Yeah.
And we've explored the incredible potential of this Schapendoes case to advance research and treatment for NCL6, both in dogs and humans.
Right.
It's a story of scientific discovery.
It is.
Hope. And the powerful bond between humans and animals.
Absolutely.
So much to ponder.
Yeah.
But for now, we'll leave you with this thought.
Okay.
How might further investigation of this Schapendoes NCL model help researchers develop effective treatments? Or even a cure for NCL in humans?
That's the million-dollar question.
Keep those minds inspired, hearts light and tails wagging from Vet Neurojar.